welcome back to the lecture series in bioelectricity. So, this is lecture number 19. So, in the previous lecture we talked about the basic structure of the retina and the eye, and there we talked about the complex layer of different cell types in the retina, which ensures that the image which is formed on the retinal plate is conveyed correctly to the brain and the decoding takes place. So, our eyes, human eyes can distinguish light wavelength from the range of 400 to 700 nanometers. Okay. So, if you recollect your previous lecture where I have shown on the retina, on the bed of the retinal pigment epithelial cells, you have these rods and the cone cells. So, the rods are the ones which could distinguish the different intensities of light, whether it is in during the dark conditions or during heavy light conditions likewise, and the cone cells are the ones which could distinguish the different colors. So, essentially cones are of different types. It could be a red cone, it could be a blue cone, it could be a green cone, depending on the specific ability of that cone cell to distinguish a specific color type. Okay. So, today what we will do, we will be talking about the electrical activities of these rods and the cones under one broad heading, the photoreceptor, electrical activity of the photoreceptors. And then we will be talking about the current stage of the prosthesis where people have lost their complete vision or partial vision. So, essentially there are two kinds of blindness as I have previously described. Either the blindness could be at the level of the lens, where there is a damage on the lens and the lens has to be replaced by an artificial lens. Those are curable blindness, but there is another level of blindness which arises at the level of the photoreceptors, where the photoreceptors gets damaged either because of old age or because of some birth defects or because of some injury or some other pathophysiological situations. So, let us first of all discuss the structure of the rods and cones, and then we will talk about the electrical activity in terms of the dark currents which are generated and why these are called dark currents, and then we will briefly talk about some specific aspects of the cones, and then we will talk about the prosthesis. Okay. So, to start off with, so we are into lecture 19. So, basically we will be dealing with the photoreceptors, and within the photoreceptors we have the rods and the cones and their electrical activities. The concept of the dark currents, and then followed by that we will be moving on to the prosthesis. Okay. So, moving on to the next slide. So, let us talk about the structure of the rods and the cones. So, these are very unusual kind of a structure. These are almost like fairly elongated structure like that, and if you could see a cone, pretty much a cone looks like this. So, this is the part, say for example, if you look at this pen, so you could see that there is this kind of light black color or ash color body, and there is underneath there is a lighter color. Okay. So, this is the part essentially where all the light receptive pigments are present, and this is the cell body. So, if I draw it, it looks something like this. So, for example, cones, because of their whole structure, they are more like a structure which similar to this, and this is the classic structure of a cone like cell, and within this structure you have these what do you see, this cap like structure what I have just now drawn for you. So, this is where all the light sensation or different wavelength distinction takes place, whereas if you look at the rod, they are more like more almost like a rod only. And here you have okay. So, this is where, so they are called cone, they are for the color perception. So, these are the cones here for the colors, and these are the rods for different light intensity. Okay. So, this is where they have the nucleus, and the way it works is, this is where the light falls. This is the one, the cone is the one which could distinguish between, the green, it could be a green cone, it could be a blue cone, or it could be a red cone, almost like, R, G, B coding. Okay. So, the different wavelengths of these, this could distinguish. Yeah. So, these are the different wavelengths. So, the specific cones are of a specific type. They could be excited, so that they could emit, they could absorb light at the blue wavelength, or they could absorb light at the red, or they could absorb the green wavelength. Okay. So, for example, this kind of color vision is only functional in high intensity light, because if you walk in the dark, you will realize that you cannot distinguish color. What you essentially see a shade of black and white. So, that means the cones are only active when you are in a bright light, and where you could distinguish the different color, red, green, likewise. And there are people who suffer from color blindness, where they could not distinguish this different kind of color. So, that means such individual suffers from certain genetic disorders, where their color distinction, the specific type of cones which could distinguish color, does not develop properly, or maldeformed or malformed. Okay. So, there is another kind of blindness which is called a night blindness, which is essentially, as it becomes darker, you are unable to really distinguish objects properly. That essentially happens because of the rods, and it is not a genetic disorder, it is mostly because of the malnutrition, of lack of vitamin A in the food, and we will come to that, where it exactly happens. In the case of rods, if you look at this structure, so out here any kind of, like, the lights which are falling here, and they respond to the light. So, essentially what happens when the light falls, so these light or these photons binds to specific sites on the membrane, and once they bind to the membrane, this light energy is translated into an electrical impulse. This is what essentially happens. So, here you have the light signal falling on photoreceptor, which includes rods and cones. Depending on the situation, it could distinguish the color component, and if it is dark, it is not going to do so, and the intensity, and these are coded as electrical signals. Electrical signals, and these electrical signals are essentially sent to the brain for further processing. And in between the rods and the cones are the whole network of amacrine cells, horizontal cells, which ensures that the exact depth of the object what we are looking at, the exact the movability, and all these features are being added to this. So, what the brain receives is a very complex electrical signal which it has to distinguish in terms of colors, intensity, depth, frame, and all those things. Okay. So, coming back to the structure. So, how these electrical signals are being received. So, it is very interesting that on the photoreceptor there is one term which I am going to introduce now is called dark currents. So, what does dark current means? So, there is a common way we look at it. So, we believe when there is an intense some signal is coming, our sensory cells respond to that and generate action potentials. Okay? And this action potential eventually is conveyed to the brain, and we decipher the object or whatsoever we are looking or we decipher the information. Right? In the case of rods and cones, this thing is just reversed. It's something like that. When the light falls on the photoreceptor layer, they do not generate any signals, and when the light does not fall, they generate signals. It is just the reverse. It means when the light is not falling, suppose my eyes are closed, so it is actually conveying the signal. So, essentially what happens, suppose you have a matrix like this you have to conceptualize in your brain, then only you will be able to understand. So, for example, this is the matrix of say for example, these are different matrix of say rods and cones which are sitting there. I am just putting different colors for your understanding. Okay. Okay. So, imagine this is a photoreceptor layer, and these individual circles are different photoreceptors. Okay. So, say for example, let me just okay. Give me 1 minute, extra thick. Okay, fair enough. When the light is falling here like this, and this is the wave of light which is falling. So, when the light is falling, differentially at some point this will, one second. Okay, at one point this one will get shut off, then at the next point this one will get shut off, and this one will get shut off, this one will get shut off, this one will get shut off. But this all shutting off is taking place, if this is taking place at T1, this will take place at T2, this will take place at T3, T4, T5, likewise. I am just randomly putting all the different numbers at different, T stands for the different time. So, essentially what is happening, you are getting a matrix over a period of time, where initially this one switched off, there is one frame you are getting where one, say for example, think of a floodlight. One light goes off, there is one image, next light goes off, you get next 3 light goes off, you have another image, next 4 light goes off, you have another set of image. And essentially what is happening, you are recording this frame by frame, and eventually the whole image is formed in front of you. Or say for example, in a city, you are looking from the top, all of a sudden I switched off all the lights in such a way that you make a figure. So, I just selectively put certain lights on and switch off all the lights, and then I change the figure. I again switched off certain lights and gets a figure. Likewise, I can really frame by frame I can actually convey the message. This is exactly how some of these the retina actually, this is kind of an analogy how it works. So, for example, at the frame 1, when if you talk about the frame, let's try to see how let's draw 6 frames what is happening. Okay. So, this is say for example, time T1, this is time T2, this is time T3, this is say T4, this is T5, this is T6, and this is, let us assume that this is our. Okay. So, when the first shot of light comes, this one goes off. Okay. The next frame, so for example, this one, and this one goes off. So, now between these two you will see initially you saw a image like this, what I am circling, something like this with a contrast here, there is a contrast here. Then you see an image like this, with a contrast here. You are kind of seeing a shape now. Okay. Likewise, frame by frame, and this is all happening within a frame of nano or femtosecond. You have lot of frames, and these frames are the ones which eventually form the image. This is very, very essential for you to understand before I go to the individual at the cellular level. So, it is a frame by frame images which are formed because of shutting off of say one photoreceptor or some population of photoreceptors, the other photoreceptor layers are active, then another set of photoreceptors is active, the other set of photoreceptor goes off, likewise. So, whenever you have to imagine, think of a floodlight in a velodrome or in a stadium, where if one light goes off, you have one frame, then few lights goes off, you have another frame, then few lights goes off, another frame. Or you can like think of it, somebody is showing you the sky, and slowly one by one, showing you the star, follow the stars. It is almost like that, one switch off, switch on, switch off, switch on, likewise. Okay. Something of that. Or suppose you are seeing a city from the top, all the lights are on, then somebody selectively switches off a light and make a figure, then switches of the light makes a figure, switches of the light make a figure, and then that whole image will come. Exactly that is how the retina functions. Now, coming at the cellular level, what is happening? Okay. So, I can use this. Okay. So, what we can do, what I will be doing actually, now I will give you the membrane structure here. Now, let us see the structure of the membrane. So, we will be talking mostly about the rods at this time. Rods, I mean, the signal processing is fairly the same. So, okay, so here you have the rods. So, if you really blow up this image, it is almost like this. This is the membrane structure, which remember in one of the previous lectures we have talked about the membrane. This is the lipid bilayer, and on the lipid bilayer you have something like a protein sitting there, which is called rhodopsin protein. Okay. The rhodopsin is a huge molecule, rhodopsin, which is essentially made up of two. One is called opsin, the other one is called retinal, and the retinal fragment is sitting somewhere. I will just put it in blue now. Somewhere here the retinal fragment is sitting. Okay. So, this is how it looks like. So, there is an opsin moiety and there is this retinal molecule. So, this is the key molecule which ensures, or this is the one which receives the light. So, it receives the light. So, this has a light, this is a light sensitive protein. It is this molecule which ensures, it has variants of course, which ensures that at different intensities how it will function, with different wavelength how it is going to function. So, this key molecule, now we will be talking about what exactly happens with this molecule in the next slide. The next slide what I will do, I will talk about. So, let us again get back to the structure of, let us take the example of a rod now. So, here you have all those rhodopsin molecules which are sitting here, and okay. Here you have the nucleus, and now say for example, you put an electrode inside the cell, and you put another electrode of course outside the cell like this, and you have voltmeter sitting here. What will you see? So, initially what will you see when there is no light falling in this? So, at that time your voltmeter reading will tell you around minus 40 millivolt. Okay. This is a situation when one second, there is no light. Let we put that, there is no light, and at that point there is a flux of the sodium which is getting in. As the sodium is getting in, it is ensuring, all the sodium ions are ensuring that these electrical impulses are being transmitted. So, this is the kind of the axonal end of a rod or a cone cells. Okay. So, because when the light is falling, light is not falling, sorry. So, mark it, light is not falling, there is profuse entry of the sodium ions, and this ensures that it generates action potentials which are transmitted to the next layer, what you could see. So, at this stage the electrical signal is traveling like this. Now, when the light falls, so this is basically what I termed as, what I was trying to discuss with you is the dark current. There is no light, yet it is generating action potentials. Now, next let us go to the next situation. In the next situation, what is happening? Here you have again the rod cells. Okay. Now, again the same, okay. You have an electrode inside, you have the voltmeter sitting here, and you have the other end of the voltmeter. Now, light is falling on this. When light is falling on this, what you see, the membrane potential goes to minus 70 millivolt. Now, what is happening here? When minus 70 millivolt is out here, it means it is no more sending any signal out here. Just reverse to the situation where during no light it was sending signal, and you see without light. So, basically the signals are being, there is no signal. What exactly happened? This is a very, very interesting phenomenon. So, let us dissect the problem at different level. First of all, when the light was falling, sodium channel was not falling, sorry, excuse me, when the light was not falling, sodium channels were open. When the light started falling, sodium channels were closed. So, what is regulating the sodium channels? First question. And how that regulation is modulated by light, because when the light falls, sodium current stops. So, it means light and the gating of sodium channel is linked. So, these are the two things which now you will be discussing, light and gating of sodium channels are linked. And now, what we will do, we will establish that linkage, what essentially is happening. Okay. So, there are two things which are happening out there. So, the first thing what happens is, now let us go back to the membrane structure, what is happening. You remember, so here is the rhodopsin molecule, and here the lipid bilayer rhodopsin molecule is sitting out there with its opsin moiety and the retinal moiety. So, I told you that somewhere here is the retinal fragment is sitting, okay. And this is a protein which is with all its complex structure out there, which is shown in green. Okay. Now, when the light falls, so this moiety, what you see here, this retinal moiety, this remain as 11 cis retinal with an aldehyde group. Okay. This is in this stage, and of course, you have the once again opsin moiety, and altogether it is a rhodopsin. So, now the light is falling. At this stage there is no light. Now, here the yellow thing what he was showing by H nu is that when the light is falling. When the light is falling, what happens, this 11 cis retinal what you see here transformed into transretinal. So, the cis bond becomes a trans bond when the light falls on it. So, now what do we see? There is a change in the molecular architecture of that retinal molecule. What are the consequences of that change? This is the first change. So, within the rhodopsin, so rhodopsin is having this opsin and the retinal moiety attached on it. It's a complex protein. When the light is not falling on it, the retinal moiety remain in 11 cis retinal form. When the light falls on it, 11 cis retinal become 11 transretinal. But then what happens next? Okay, let's move on to the next slide. One second, let me save it. Okay, so the next thing what happens is this. Once the 11 cis retinal is formed, so let's go back. So here the photon is falling on. Okay, here is the moiety sitting there. So photon has formed. Now you have 11 transretinal. This 11 transretinal then dissociates from this structure. So what you are left with is now you have this opsin protein coming out plus you have 11 cis 11. One second, let me just rub it off. Okay, and you have this 11 transretinal. Okay. Now this opsin protein now remain, it's pretty much orphan at this stage, and whereas 11 transretinal through an enzymatic action again become cis. Okay. So, there is a complete enzymatic action where ATP is being used to convert it into 11 cisretinal. Now, this cis form again assemble with the orphan protein or the orphan component, which is the opsin, and again form this structure. So, this is basically what is being called is a bleaching phenomena. There is a kind of continuous bleaching taking place within the eye. Okay. But during that process, something else has happened. That is what we are going to discuss, how that channel remains open. So, what essentially happens in a step 1, if we come back to step 1 was opsin activation, because when the light falls, I told you the opsin comes out and the 11 transretinal dissociates. Okay. Then what the opsin does, the step 2, opsin does something very interesting. Opsin activates a second enzyme called transducin, and this transducin, which in turn activates another enzyme called phosphodiesterase, and this phosphodiesterase then does something very interesting. What it does is, now just before that, let me talk about a little bit about the transducin. What is exactly, just give me 1 minute. Okay. Transducin. So, transducin is basically a G protein. Okay. It is a series of proteins. G protein is a membrane bound enzyme, and it activates by interaction with receptor proteins bound in the cell. Okay. Bound in the cell membrane. In this case, what transducin does, transducin is of course activated by opsin, and transducin in turn activates your phosphodiesterase, what I have talked about. And phosphodiesterase what essentially does is very interesting. This phosphodiesterase disintegrates a molecule called cyclic GMP. then what is the role of cyclic GMP? Okay. So, coming back to the slide. Okay. Now, you have phosphodiesterase I am showing by PDE. This PDE then acts on cyclic GMP. So, what is the role of cyclic GMP in this whole context of things? What I was trying to tell you. So, let us come back to the first question here out here. What is the question what we ask? One second, let me go back to that slide where I talked about the dark current, introduce you to the dark current. Yes, when there is no light, I showed you this picture. Okay. When there is no light, you could see the sodium channels are open. Okay. Up to this concept is clear. Then what essentially happens when the light falls? Sodium channels become closed. So, the fundamental question comes here. We asked two fundamental questions. One was this one, light and gating of sodium channels are linked. We will take it up from here in this slide. Okay. Let us see. So, light, two situations, no light, sodium channel or sodium current, sodium channel closed, okay. No light, sodium channel open, fine. Now, the next challenging question was, what keeps this sodium channel open at this stage? As if a door is kept open. So, how you keep your door open? There are three ways how you can keep a door open. Think of the room where you were sitting and listening to this lecture. You can keep the door open by using like something underneath the door stopper, or you have something by which you keep the door like this. So, these molecules have something like a door stopper molecule, and the door stopper molecule or door opener molecule of sodium channel is cyclic GMP. In other words, what is essentially is happening, if say for example, this is, let us take an analogy that this is the door of a room, this is the wall and this is the door which is locked. So, there is cyclic GMP, if this is say for example, the sodium channel and this is in a closed state, okay, or this is the door. Now, this could be kept open like this by holding it with something like this. You have to hold it. It could be a door stopper or anything. In the case of channel, here you have this cyclic GMP sitting. So, cyclic GMP acts as a door stopper, keeps the door open. Okay. So, now the door will only close when you remove that door stopper. That door stopper cyclic GMP is being removed by the molecule called phosphodiesterase, which is activated by transducin. Transducin is further activated by opsin, and opsin is disintegrated in the presence of light. Does it make sense? So, when the light falls, so let us summarize what exactly happened. Okay. Now, coming back to the next slide. So, light falls on the retina. What you essentially see? No current and no sodium flux. So, it means sodium channels are closed. So, what is happening at the molecular level during this whole thing? Light falls, 11 cis retinal become 11 trans-retinal on the rhodopsin molecule. 11 trans-retinal dissociates out, and this process dissociates the opsin molecule. Okay. This opsin molecule then activates a protein called transducin. This transducin further activates a molecule called phosphodiesterase. Phosphodiesterase then dissolve the cyclic GMP, which was keeping the sodium channel in open state. This is exactly what is happening in this whole process of signal transduction within the rods and the cones, where when the light falls, the electrical signal ceases, and when the light is not falling, the electrical signal passes on. And that is why this is called a dark current, which is also called cyclic GMP regulated current. And depending on the wavelength of the light, in the case of cones, these electrical currents are being stimulated or stopped. So, this is a very fundamental concept which I wished or wanted that you people should be clear to you. It is this layer which is the most critical one. So, say for example, now somewhere or other, if this layer, so let us go back and see the global picture out here. So, out here, one second, let me just give me 1 minute. Okay. So, here you have the brain, spinal cord. Okay. Here you have the eyes. So, the electrical signal moves like this, left to right, right to left. Okay. Now, at this level, the first level where the image processing takes place is the PR or the photoreceptor layer, which is essentially the rods and the cones. So, the electrical signals are traveling from the eyes all the way to the brain, and the processing takes place. Now, imagine somewhere or other the rods and the cones layer goes bad. What will happen? So, now there will not be any further image processing taking place. You will see it, but there will not be any further message going on. This is all blocked. What intervention can happen? So, one option is that if you could replace these eyes with a camera or with two cameras on two sides. So, you have a goggles like this, something like this, where you have the cameras sitting out here, and this goggle is connected, interfaced directly to the brain. So, camera, instead of this, now they are directly cross talking with the brain. This is the approach which inspired people for last 1 century, that could we really bypass the whole vision by putting cameras instead of eyes. So, basically what is happening, this circuit is no more functional. This is a defunct circuit out here. So, what you are doing, you are interfacing the camera with the brain. So, images which are forming, analog images, are digitized, and the digitized signal in the form of electrodes are being fed to the brain. So, in this context, I will recommend you to read some of the papers, and of course, at the end of the course, I will be providing all the references which I wish. So, one of the person who had made really good contribution is from University of Southern California. You should read is Mark Humayun. Spelling may be wrong, sorry, Mark Humayun. His paper from University of Southern California, USC. He has made some very interesting cameras where he has interfaced them with the brain, and this is really a very interesting piece of work which is going on. So, where the prosthesis is kind of a successful process, but there is a lot of improvement which has to be done. So, what I will be essentially doing, I will be giving you these references of Markov ion, and I expect you to read through them, because that is where lies most of the future of prosthesis. So, again, to tell you what is the major challenge is this game. In this game, the major challenge, how we interface the electrodes with the brain, because you are putting some foreign material in the form of electrode. So, that has to be biocompatible, and not only it has to be biocompatible, it has to be ensured that all the signal over a period of time continuously reaches, because you cannot continuously open the system and fix the electrodes. It has to be done. So, say for example, here essentially what we are talking about, if this is the individual and if this is the brain. So, that is what you are saying is essentially this person has this goggles which they are putting. So, here is the retina which is receiving the light, and this goggle which is a synthetic retina is essentially interface with the visual cortex out here in the brain. So, there is enormous amount of electrode implantation which has to be done to ensure all the message reaches the brain at the visual cortex, and then of course, this person will be able to visualize if this person is unable to see. But this needs not only, I mean, of course, the camera technology is very well advanced, but the interface technology, which is the whole area of neural engineering, or you can talk in a much more specific way at the cell electrode interface or neuron electrode interface. This is in terms of the cell here. This is a very challenging area, where lot of research is going on for last 1 century, that how really we can develop extremely high biocompatible material which will convey the signal and help in this prosthesis business. Okay. So, I will close in here with this, and in the next class we will talk about the ear and the ear prosthesis, and so on and so forth. We will finish this whole prosthesis part with the basic structure. So, there also will be following the same strategy. Okay. So, and I will be adding all these references for you, and please kindly go through some of the work on Markov ion. That is very inspiring pieces of work which is happening across the globe. Thank you.